All right, welcome. This is the Flow Cytometry Basics Part 1 workshop. That is a webinar. Thank you for joining me today for our continuing Flow Cytometry Reagent webinar series. This is the first of two webinars on the basics of flow cytometry. We have two previous webinars that are available on our website for viewing. My name is Jolene Bradford, and I am the Research and Development Associate Director of Flow Cytometry Systems, which is located in Eugene, Oregon, the home of molecular probes. I'd like to make a few announcements. First, you may submit your questions at any time during the presentation. I'll try to answer as many of these questions as time allows at the end of the presentation. Due to time constraints, I may answer some of the questions after the webinar has ended. As I mentioned, today's webinar is part of a series that will continue throughout 2012. And our second webinar on flow cytometry basics will be given on March 13th. I'd also like to let you know that on April 17th, we will be presenting a webinar highlighting microbiology in flow cytometry. So let's begin. Today we'll cover the basics of fluorescence, go over components of a flow cytometer, and discuss some of the basics of data. So let's start with fluorescence. A fluorescent compound absorbs light, which causes an electron in the compound to be raised to a higher energy level. The excited electron quickly decays, emitting excess energy as a photon of light. The emitted light is called fluorescence. There is an absorption spectrum, which is the wavelength over which a fluorescent compound can be excited. There is emission. This is the range of emitted wavelengths of a fluorescent compound, and it is longer than the wavelength of the absorption. And then there is a Stokes shift, and this is the difference between the excitation and the emission wavelengths. So I'm going to go over each of these in a little more detail to allow a better understanding. So a fluorophore is a molecule that is capable of fluorescing. In its ground state, the fluorophore molecule is a relatively low energy but stable in configuration, and it does not fluoresce. When light from an external source hits the fluorophore molecule, the molecule can absorb the light energy. When the light energy is sufficient, the molecule reaches a higher energy state called an excited state, and this process is known as excitation. Since the fluorophore is unstable at a high energy configuration, it eventually adopts the lowest energy excited state, which is semi-stable. The length of time that the fluorophore is in the excited state is called the excited lifetime, and it lasts for just a very short time. Next, the fluorophore rearranges from the semi-stable excited state back to the ground state, and the excess energy is released and emitted as light. The emitted light is of lower energy and thus a longer wavelength than the absorbed light. This means that the color of the emitted light is different from the color of the absorbed light. This cyclical fluorescence process can be summarized as first, the excitation of a fluorophore through the absorption of light energy. Second, a transient excited lifetime with some loss of energy. And finally, return of the fluorophore to its stable ground state, accompanied by the emission of light. The emitted energy is always of a longer wavelength than the energy absorbed due to the energy lost during the excited lifetime. Wavelength is the distance that light travels in one vibration cycle. This distance is expressed in nanometers. The visible spectrum is composed of light with wavelengths ranging from approximately 400 to 700 nanometers. Light waves with shorter wavelengths have higher energy frequency, and wavelengths with longer have lower frequency and a lower energy. As stated before, an excited fluorophore emits lower energy than the light is absorbed, and therefore there is a shift along the spectrum between the color of light absorbed by the fluorophore during the excitation and the color emitted. So how is the excitation of a fluorophore determined? So for example, let's say we have a tube that contains a particular fluorescent dye. If we shine 480 nanometer light at the dye solution, some of the fluorophore molecules will become excited. However, the majority of the molecules are not excited by this wavelength of light. If we increase the excitation wavelength to 520 nanometers, more molecules are excited. However, this is still not the wavelength at which the proportion of the excited molecules is maximal. For this particular dye, 550 nanometers is the wavelength that excites more fluorophores than any other wavelength of light. And if you go to wavelengths longer than 550, the fluorophore molecules still absorb energy and fluoresce, but again, in smaller proportions. So the range of excitation wavelengths can be represented in the form of a fluorescence excitation spectrum, which looks like this. So how is emission determined? Well, when we excite the previously described dye solution at its excitation maximum, 550, we find that light is also emitted over a range of wavelengths. Although the fluorophore molecules all emit the same intensity of light, the wavelengths and therefore the colors of the emitted light are not homogeneous. Collectively, however, the population fluoresces most intensely at 570 nanometers. So based on this distribution of emission wavelengths, we say that the emission maximum of this fluorophore is 570 nanometers, and the range of wavelengths is represented by the fluorescence emission spectrum. To summarize, fluorophores are molecules that, upon absorbing light, can reach an excited state and emit light energy. The three-stage process of excitation, excited lifetime, and emission is called fluorescence. Fluorophores absorb a range of wavelengths of light and also emit a range of wavelengths. Within these ranges, the excitation maximum and emission maximum can be determined, and these are different. So a fluorophore is excited most effectively by light of a particular wavelength, and this is called the excitation maximum for the fluorophore. And fluorescent emission behaves in a similar way. The fluorescence output of a fluorophore is most likely to occur at a particular wavelength, and this is the emission maximum for that fluorophore. It's important to remember that although illumination at the excitation maximum of the fluorophore produces the greatest fluorescence output, illumination at lower or higher wavelengths affects only the intensity of the emitted light. The overall range and shape of the emission profile are unchanged. So the excitation source, just remember that the fluorescence emission is always longer than the excitation wavelength. In this example, using 488 nanometers for the excitation source, the emission range will always be of a longer wavelength or greater than 488. The emission maximum for the fluorophore, since it is always at a longer wavelength, that is a lower energy, than the absorbed light of the excitation wavelength, there is a difference between the wavelengths of the excitation and the emission, and this difference is called the Stokes shift. So here's an example of the excitation and emission spectra of Alexa Fluor 488. This is a commonly used fluorophore, and this contains very practical information about the wavelengths of light that we need to supply and detect in order to use the fluorophore effectively. The excitation and emission spectra must be examined carefully when choosing two or more fluorophores to use simultaneously in an experiment, so that the fluorophores can be excited in a manner that will generate distinct emissions. Spectraviewers like the one offered on the Life Technologies website are very helpful for this. In this example, we see the excitation spectra of two fluorophores, alexafluorofluor 88 and alexafluor 647, are shown, along with the two laser lines, 488 and 633 nanometers, that are used to excite them. For your convenience, there is also a Spectraviewer mobile app available for free download. So in the Spectraviewer, you can build on the example, and we see that there are emission profiles with collection band filters that can be added. And in this example, we see Pacific Blue and Pacific Orange. These are two fluorophores that are commonly used with the violet 405 excitation source. And you can see that they are optimally excited by this laser line. And then the emission profiles are shown with the typical bandpass filters that are used for collection. So we have our first polling question. This is a true or false question, and you can answer as soon as you're ready. True or false? Fluorescence emission is always longer than the excitation wavelength. This is a true or false question, and you can vote right now. Fluorescence emission is always longer than the excitation wavelength. So I'll give you a few more seconds to respond. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. And I can see that of the respondents, we have about 80 people have responded, and 96% of you have correctly identified the answer as true. The emission wavelength is lower in energy and thus longer than the excitation wavelength. Okay, so now let's move on to some of the basics of the flow cytometry technology. Cytometry is the measurement of physical or chemical characteristics of cells or particles. Flow cytometry measurements are made as cells or particles which are in a suspension pass individually through a flow cytometer. This provides discrete measurements from each cell and provides a distribution of the measured characteristics. Flow cytometry is dependent upon the control, the collection, and the separation of light. This technology simultaneously measures and then analyzes multiple physical characteristics of single particles, which are usually cells, as they flow in a fluid stream through a beam of light. Fluidics introduce the cells for interrogation with the laser. Optics generate and collect the light signals. And then finally, electronics convert the optical signals to the proportional electronic signals for computer analysis. There are many advantages of flow cytometry. This records the data from single cells. This makes measurements on large number of cells, generally in the thousands to millions per sample. Rich statistical analysis can be obtained on populations. And because single cells are measured, it will reveal the heterogeneity or the diversity within a population. There is the ability to multiplex. Small subpopulations can be identified. Cells are rapidly analyzed, often within seconds. This is ideally suited for blood and other cells in suspension. There is the ability to archive the flow cytometry standard file, which is called FCS file. And because of this, the data can be reanalyzed at any point in the future. There are some specialty instruments that are capable of sorting. These are called sorters, and this is different than an analyzer. and subpopulations can be collected for further study. And there are other cytometers that can be used with imaging at the same time, giving morphology and spatial information as well. The cells or particles in suspension flow single file through a laser beam where they scatter light and emit fluorescence. And the fluorescence is collected and converted into digital signals where it is then stored on the computer file. The purpose of the Fluidics system is to transport particles in a fluid stream to the laser beam. The sample is injected into the stream of sheath fluid. For optical illumination, particles or cells should be at the center of the laser beam. Only one particle should pass through the laser beam at one time, and the Fluidics systems need to be free of air bubbles and debris. In the flow cytometer, particles are carried to the laser intercept in a fluid stream. The portion of the fluid stream where the particles are located is called the sample core. When particles pass through the laser, they scatter laser light. Any fluorescent molecules that are present on the particle will emit fluorescence. The scattered and fluorescent light is collected by lenses to steer the light to the appropriate detectors. One of the most common ways of focusing the particles is using hydrodynamic focusing. In this method, the sample is injected into the stream of sheath fluid where it is completely surrounded by the sheath fluid itself. The sample core is then centered within the sheath fluid where the laser beam will interact with the particles. Based on principles relating to laminar flow, the sample core remains separate but coaxial with the sheath fluid. The flow of sheath fluid accelerates the particles and thus restricts them to the center of the sample core. And this process is known as hydrodynamic focusing. Where the sample core is narrow with a low flow rate, the particles interact with the laser beam optimally, and a narrow distribution results as seen in the histogram above. This is typically about 12 microliters per minute. To increase the flow rate in a hydrodynamic system, the speed of the sample core does not change, but rather the width of the core within the sheath fluid increases, which allows more cells to enter the core stream at a given moment. With a wider sample core, some cells will pass through the laser beam off-center and intercept the laser beam with a less than optimal spot, creating variability of signal. This results in a wider distribution of data, and resolution is decreased. This limitation, the trade-off of speed and sensitivity, has largely been accepted by flow cytometry, and this requires the use of low flow rates for samples where a very precise result is required. A typical high flow rate is about 60 microliters per minute. So here's another way to visualize some of the trade-offs seen in changing from a low to a high flow rate with hydrodynamic systems. And again, we're showing a range of 12 microliters per minute for a low flow rate, and at a high flow rate at around 60 microliters per minute. More recently, a newer method of focusing has been introduced. This is acoustic focusing. With this method, regardless of the sample volume that is moved through the flow cell, cells are focused by an acoustic device prior to entering the flow cell. So at low sample volumes, as seen on the left, and high sample volumes, as seen on the right, focus is precisely maintained. This also means that very little sheath is needed. It is only used to allow varying the amount of sample while maintaining the correct flow rate, and it is also used to help clean or help keep the inside of the cuvette clean. So this is another representation of acoustic focusing, and because of the precise alignment of the cells in the middle of the sample stream, no matter how wide the core is, this does allow for collection rates much higher, and in this example, we see 25 microliters up to 1,000 microliters per minute, which allows for much faster collection rates than a hydrodynamic system. And this is shown with the acoustic the Attune acoustic cytometer, which has been designed and powered by molecular probes. Excitation optics create an illumination region. They include lasers and lenses to shape and focus the laser beam. Collection optics route the generated light signals to the specific detectors. They include collection lenses to collect light emitted from the particles and include a system of optical mirrors and filters to route the specified wavelengths of light to the designated optical detectors. Laser stands for light amplification by stimulated emission of radiation. It is a coherent light used by most flow cytometers to excite fluorescent dyes and characterize the particles as they pass through the laser beam. Here we see some of the most commonly used laser excitation sources. This is not an exclusive list. There are others which can be used, such as 550 or near IR wavelengths. Light scattering occurs when a particle deflects incident laser light. The extent to which this occurs depends on the physical properties of the particle, namely its size and internal complexity. When a cell passes through a focused laser beam, it refracts or scatters light in all directions. We typically look at forward and side scatter light. Forward scatter, or low-angle light scatter, is light that is scattered in the forward direction as the laser light strikes the cell. The magnitude of the forward scatter is roughly proportional to the size of a cell or particle, but it's important to note that forward scatter does not give an absolute particle size. It is not a volumetric measurement. Side scatter is defined as the light that is scattered at a 90-degree angle. This is indicative of granularity and structural complexity inside of the cell. Forward and side scatter measurements can be combined in a dual parameter plot, such as these, to allow differentiation of cell types in a mixed cell population without any fluorescence. This is just scattered light. Optical filters separate the light scatter and fluorescence directed to detectors by wavelength. They selectively transmit light having a particular range of wavelengths while absorbing or reflecting the remainder. There are 5 types of optical filters used in flow: the bandpass, long pass, short pass, dichroic, and neutral density filters. The bandpass filter is a device that passes wavelengths within a certain range and rejects wavelengths outside of that range. The bandwidth of the filter is simply the difference between the upper and the lower cutoff wavelengths. Common bandpass filter nomenclature uses the peak emission / bandwidth. For example, a filter that would detect the Alexa Fluor 488 dye would be a 530/30, and it is labeled according to the middle wavelength of the band, which is 530, and then passes only a narrow band of colors in the wavelengths 515 to 545. A long pass filter is an optical filter that rejects shorter wavelengths and transmits or passes longer wavelengths. In this example, a long pass 530 permits wavelengths longer than 530 to pass while reflecting or absorbing wavelengths shorter than 530. A short pass filter is an optical filter that rejects longer wavelengths and transmits shorter wavelengths. In this example, a short pass 530 filter allows wavelengths shorter than 530 to pass while reflecting or absorbing wavelengths longer than 530. A dichroic filter can also be called a dichroic mirror. These are produced by coating a glass substrate with a series of optical coatings. Dichroic mirrors usually reflect the unwanted portion of the light and transmit the remainder. And finally, there are neutral density filters, which have a constant reduction across the range of visible wavelengths, and they are typically used to reduce the intensity of light by reflecting or absorbing a portion of it. So here's an example of a very simple optical setup. This is using the laser and filters to direct the light to the collecting devices, here labeled as PMTs, or photomultiplier tubes. This is a simple diagram showing the fluorescence collecting at 90 degrees with 4 detectors and 1 laser. Here is a different configuration. This demonstrates a more complex pathway using 2 lasers, 2 scatter parameters, and 6 fluorescent detectors. In most multi-laser systems, each cell passes through each laser interrogation point at different times. The information obtained at each interrogation point is aligned in time so that it is recorded at one time for each cell or each event. This coordination is accomplished using a laser delay. Some systems are collinear and do not require a laser delay method. Electronics convert optical signals to proportional electronic signals, or voltage pulses. We can analyze the voltage pulse height, area, and width, and we also interface with the computer for transfer of data. The light signals are generated as particles pass through a laser beam. These light signals are converted into electronic signals, or voltages, by photodetectors, and then are assigned a channel number on a data plot. There are generally 2 types of photodetectors in flow cytometry: photodiodes and photomultiplier tubes, or PMTs. The photodiode is less sensitive to light signals than the PMTs, and it is often used to detect the stronger forward scatter signal. PMTs are used to detect the weaker signals generated by side scatter and by fluorescence. When a cell or particle passes through a focused laser beam, it refracts or scatters light in all directions and can also emit fluorescence. The scatter and fluorescence last only a few microseconds because the particles are moving very quickly. The detectors convert this momentary flash of light into an electronic signal called a voltage pulse. When the cell begins to enter the laser beam, the signal intensity is low because only a small portion of the particle scatters the light. As the pulse reaches its maximum, when the cell is in the middle of the laser beam, the whole particle scatters the light. And finally, as the cell or particle exits the beam, the signal starts decreasing and eventually trails off below the threshold. So this data is further processed to simultaneously calculate the pulse height, the pulse area, and the pulse width when the pulse exceeds the user-specified threshold values. The signals that the forward scatter, side scatter, and fluorescence sensors generate are proportional to the intensity of the light that the sensor received. So now we're at our second polling question. This is another true or false question. So you can start submitting your response now. Forward scatter is an absolute measure of size. True or false? Forward scatter is an absolute measure of size. And I'll give you about 10 seconds to submit your response. 9, 8, 7, 6, 5, 4, 3, 2, 1. All right, so we have 83% of the responders correctly identified this answer as false. Forward scatter is not a volumetric measurement, and it can only be used for a rough estimate of size. Okay, so let's move on to some basics of data. The flow cytometry standard, or FCS, is the format that is used to save data. All software vendors use it when creating files so that the files can be analyzed across different software packages. The name list mode file is sometimes used because the data are acquired as a list of values for each parameter from each detector, whether it's scatter or fluorescence. And this is for each event or each cell. currently we are using FCS 3.0, but there is movement to the newer 3.1 format. The flow cytometry standards files use keywords. These generally describe instrument settings and user-entered information. And this is placed into the FCS file by the data acquisition software. I have included a website here that more fully describes the flow cytometry standard files and keywords for those of you who may be interested in learning more about this. So emitted fluorescence intensity is proportional to the binding of the fluorescent label or marker. In this example, I have a 50 labeled particle with varying amounts of binding. As there is more binding of the fluorescent probe, the intensity along the x-axis increases. A histogram is a graphical representation of a single parameter data and shows the relative number and distribution of events. In a histogram, the horizontal axis or x-axis corresponds to the signal intensity of the selected parameter, while the vertical axis represents the number of cells or the count. Histograms can give rise to a variety of shapes as seen in the examples at the bottom of the slide. A histogram can also be called a single parameter plot. Lines can be drawn around populations in order to distinguish them from other populations and to collect data on that population. So they can be created on a histogram or another type of plot. So a dot plot is a graphical representation of two parameter data where each axis represents the signal intensity of one parameter. Each dot in the dot plot corresponds to one event detected above the threshold. A density plot is very similar, is that it's a graphical representation of two parameter data where the colors represent the collection of events with the same intensity and each axis represents the signal intensity of one parameter. Now the two parameter plots shown at the left display the same data. The one on the top shows the dot plot, and the one on the bottom shows the same data as a density plot. Two parameter plots do not have to be rectilinear in their display. As you can see on the right, this is using our Click@edu and DNA content dyes in a dot plot format. We see a typical density plot on the left where this is a rare event seen in the upper right quadrant, and most of the cells are negative in the lower left quadrant. Gates and regions are commonly used in data analysis to identify different subsets of populations in multicolor experiments. So regions are shapes or objects that are drawn around a population of interest on one or more or two parameter plots. A gate is defined when regions are used to isolate a specific group of events from a larger set of data. Gates can be customized by using Boolean logic to link multiple gates together. The most popular application is to use gates in a sequential order to limit the data display and to define the parameters for a statistical data or statistical analysis. The plots at the bottom of the slide each show a region that has been drawn around a population of interest. In this example, this is a human blood sample which the red cells have been removed through lysis, and we have a mixed population of cells. And you may want to restrict your analysis only to the lymphocytes. And here we have a region that is created around the lymphocyte gate. This becomes a gate when it is used to analyze further subsets within the sample. So here we can see statistics from each of the quadrants of the three two-parameter plots on the gated parameter. Here the statistics are displayed in a hierarchical fashion. So gating tools can be used to isolate a region in a selected plot for analysis. There are a number of different shapes that can be used. Most commonly, there is a rectangular gate. You can use an oval gate, or a polygon gate can be used to customize the shape of the region that is being used. And a common gating strategy in two-parameter plots is using a quadrant gate. And in the more modern instruments, these gates can be bended to fit a population better. Overlay plots are made by combining the data from different sample files and displaying them together on the same plot. These can be very useful for comparing results of different samples within the same experiment. Dot plots can also be displayed in an overlay format. And there are a number of statistical tools that can be used in flow cytometry, and that is one of the main advantages of flow. Some of the commonly used statistics are percent total. This is the percentage of a population of the total events collected. A percent of parent. This is percentage of a population based on the number of events collected in the parent gate. Median, or 50th percentile. This is the value that corresponds to the middle item in a ranked list of all measurements. The mean is calculated by summing the values of all measurements and then dividing by the number of the measurements. Percent coefficient of variation is percent CV. This is a measure of the peak distribution. It often gives you a measure of precision. And standard deviation, which is a measure of the difference of each run from the mean of the sample. There are a number of other different statistics that can also be used. So we have many resources that are available to you. The latest one is the Flow Cytometry mobile app. You can go to your mobile device and download it directly, or follow the link to learn more about this app. You can also find more technical resources by visiting our Flow Cytometry Resource Center. And finally, I'd like to invite you to join our Facebook pages on flow cytometry and immunology. And here you can share your questions and learnings with the community. And with that, this brings us to the end of this webinar, and I will begin answering questions that have been submitted. Thank you for your attention, and I'd like to remind you that our next webinar is on March 13th. But the second part of this webinar on flow cytometry basics, which will include a discussion of compensation. So one of the first questions here, is there any CEE documentation? And for this presentation, no, we do not have continuing medical education documentation available. Now, let me scroll through some of these questions. So there was a question about the nomenclature for Alexa Fluor dyes and what is the excitation or emission wavelength. So the Alexa Fluor dyes are, so Alexa Fluor 488 is based on its excitation wavelength, not its emission wavelength. There is a question about how small of a particle size can a flow cytometry measure. This is a really interesting question, and there is no real answer to that. It's going to depend on the specific optics and fluidics of a particular instrument. I would say that most cytometers can get to down to a particle size of around 1 micron. Some can go lower, but you have to be very careful in distinguishing the actual particle from the background debris. And this is where, if your small particle is fluorescently labeled, it really helps to distinguish that from background and debris. So there is a question around plant cell suspension, where single cell status is difficult to achieve. So you can Of course, you don't want clumps in your sample because that can potentially clog up the instrument, and of course you don't want to do that. But if you do have some aggregates in your cell suspension, you can use fluorescence pulse, height versus area, or area versus width, or area versus height to distinguish aggregate cells. And that might be a way to help eliminate some of your aggregates from your analysis. So the direction of the sheath fluid is in the same direction that the sample core is traveling. Is forward scatter proportional to cell size even with small cells? Again, it's relative only, and depending on the angle of forward light scatter, the proportionality will change. So you have to really look at this with your own flow cytometer, and one way to do that is to look at different size particles and run them on your cytometer to see where those size particles fall. So there's a question about what is the difference between side scatter A and side scatter H. So this stands for area and height. So this is the difference between the area pulse and the height pulse. And most cytometers will have an area scaling factor to make sure that these are equivalent. For most applications, side scatter, it probably won't matter whether you use area or height. For fluorescence detection, if you're using a very bright signal for DNA content, you probably want to use an area fluorescence detector. Let's see, there's a few questions here about reagents that I'll get back to the individuals about. Let me see. So there's a question about setting the voltage and selecting the voltage that gives a higher fluorescence. Selecting the proper voltage for an experiment is one of the more difficult things in flow cytometry. And my suggestion is that you have a positive signal, your control, that needs to be on scale, and your negative signal also needs to be on scale on the other end. So when you're setting up your instrument, please look at both of those and try to make the best that you can there. The voltage that you select will also have an effect on your compensation. So I invite you to come back to our next webinar and we can discuss that further. There is a question about, does an event mean one cell? And yes, that is true. An event can also mean a bead or a particle that is being measured. So there's a question about a cell cycle and looking at S phase and whether gating, whether you should gate cells or analyze it without gating. And there's a couple of schools of thought on this. So if you are using a secondary program that is using probability, I would suggest that you don't need to gate for your analysis, and you can allow the software program to do that for you. However, if you're not using a secondary program and you want to look at your cells, then I would suggest that you do look at looking at gating singlets at least, and then perhaps putting a fluorescence threshold so you're only getting the cells of interest. So there's a question about how to select the right gate and know what the population of interest is. This is going to depend on your particular experiment and understanding what the expected results are or what the hoped for results are, and really looking and understanding how the cells are interacting with the flow cytometer. So there is a question about understanding which cells are dead, and this is a really good question. For the most part, scatter detection is not a reliable method for distinguishing dead cells from live cells, and it is best determined with a fluorescent dye. There's two classes of dyes that can be used for this, the cell impermeate nucleic acid dyes, such as the Cytox dyes or propylium iodide. And there's also the live dead fixable stains, which can be used for intracellular staining. These are really reliable methods, and you can gate on your live cells for further analysis. So there's a question about the difference between a dot plot and a density plot. So a dot plot is simply a representation where each dot represents one cell. A density plot puts it more in a population. So if you were to consider the populations as they're represented in a gradient of color as kind of a mountain in a 3D format, it gives you an idea of the density of a population within that plot. So there's a question about how do different flow cytometers differ. So there are a lot of different cytometers available on the market from many different vendors. They differ in the selection of their lasers, the color of the lasers, and the number of lasers. And then within that, how many detectors there are. And of course, each company has their own unique features that they promote within those different cytometers. Some of them will offer things like absolute counts, if it's a syringe-driven, or acoustic focusing. So there's a variety of different things that different flow cytometers show. So there is a question about modulating voltage for compensation. And yes, I will be covering this in the next section. But once you do set up your voltages to run your compensation, you do need to keep them constant. So they do have an effect on the compensation values. How do you draw your gates consistently between experiments? That's a really good question. There are some instruments which allow you to save a template and have all of the gates and markers consistent, but from a day-to-day, the voltages and samples may vary. So this is something that if you have some software programs have the ability to do automated gating, and that's one way to allow for less variations from sample to sample. And other than that, it's somewhat empirical by the user. So there's a question on different colors of the laser, such as violet and yellow, and what does it represent? So these actually represent different wavelengths along the light spectrum. So when we refer to a violet laser, this refers to around 405 nanometers in wavelength. A yellow laser would be 561 nanometers. So that represents the excitation wavelength, and it will help determine what fluorophores or what reagents you can use with that system. There's a question about interference when using a strong and a weak signal. I'm assuming this means in different channels or using different fluorophores, and there can be bleed through from one channel into the other, and there is a way of helping to define your experimental design so that you pair your weak fluorophores with your strong markers, and that's something that we'll cover again next time. By exponential display, so there are 3 different types of scales. There is a linear scale, there is a exponential or logarithmic scale, and then there is a newer bi-exponential scale, and this is a combination. And this is actually very useful, and I will go over this some more with the compensation module, but when this helps with visualization of data. So when a bi-exponential plot is logarithmic, except around 0, it becomes linear. And then there's actually negative numbers that can be displayed as well. And if you've ever seen a logarithmic scale where you have events piled up on the axis, if this is moved into a bi-exponential display, which can also be called logical or hyperlog or linlog, you can actually visualize these events, and this will help determine whether or not the sample is properly compensated. So what is the meaning of area scaling? This is trying to keep the area pulse and the height pulse equivalent, and trying to get those when you have an area result and a height result, making them the same. And this is something that the software and the hardware work together to do. So there's a question about compatibility between different dyes and cell types. And I'm not sure exactly what that means, but my suggestion is that you do optimize your experiment to your cell type and the different dyes that you are using for that. Can you use flow cytometry to detect cells in different stages of the cell cycle? That's a really good question. We do have DNA content dyes that can give you a rough estimate of that. Of course, you can put those through probability software modeling programs to get a better idea. There are other dyes that can be used, such as the Fluch, which is fluorescent protein, has two different readouts depending on the stage of the cell cycle. And then there are different antibodies that can be used in conjunction with a DNA dye. So there's a question about do dead cells have more or less emission? And if you're looking at a forward scatter, cells tend to decrease in size. And if you're looking at side scatter, dead cells tend to increase in side scatter as they die. But really the best way to determine what a dead cell is to use a dead cell discriminating dye. There's a question about making a gate on a population and looking at different samples and wanting to change the gate for each sample or increasing the gate size. And I think this is something that you'll have to determine for your own experiment. But if you're in the course of an experiment, the population of interest might change. So I think that's something that is possible to do. So there's a question about the criteria used to select fluorophores with a particular flow cytometer. And this is a really great question. It comes to the heart of what a flow cytometer is, and it's a set of excitation sources, usually lasers, and different collection parameters, which are the bandpasses or the different collection optics. And you really do need to understand what those are for the particular instrument that you're going to use. And then you pair that with the spectral qualities, the excitation and the emission qualities of the dyes that you want to use. And it's a selection process, and this is where Spectraviewer becomes very useful in helping to determine that. So I would say start with your flow cytometer first and find out what the limitations and of that system are to allow you to then select the reagents that can be used with that particular cytometer. So there is a question about sorting, which I really didn't cover here. And there's you need to use beads as well as an isotype control to set up the compensation. This is really a compensation question rather than a sorting question, and there are a number of ways to set up compensation. Some of them can be used with beads. Some of them can be used with cells. You can also add an isotype in there if you'd like. But it's going to really be dependent on the cells that you are using in your experiment and how you want to set up the compensation. So again, I'll invite you to come back to our next webinar. And with that, I do see that we are at the end of our session here. There are a number of other questions that I was unable to get to because of the time constraints, so I should be able to email back answers to those questions that I haven't been able to get to. And I'd like to thank you again for your attention and invite you back for our next webinar.